Hello, good evening. Welcome again. Today we discuss megasporogenesis to fruits. First, megasporogenesis. Till last class, we discussed about development of ovule or type of ovule, or we discussed about the types of ovules. Now, megasporogenesis. Megasporogenesis normally takes place in ovules. Megasporogenesis occurs in the ovules. Every ovule shows central diploid nucellus, which is nothing but megasporangium. It shows a funicle, chalaza, integuments, micropyle, and other things. During megasporogenesis, one of the cells in the hypodermal cells of nucellus converts into a special cell called archisporial cell. This chapter is important, that's why I am discussing. This archisporial cell is diploid, which divides and redivides to produce two cells. One is parietal cell, the other is sporogenous cell. Parietal cell divides and increases the amount of nucellus. Sporogenous cell divides and gives rise to megaspore mother cells. Sometimes archisporial cell directly give rise to megaspore mother cell. So, some of the hypodermal cells or one of the hypodermal cells in the nucellus transforms into archisporial cell. Archisporial cell divides and gives rise to parietal cell and sporogenous cell. Parietal cell increases the amount of nucellus. Sporogenous cell converts into megaspore mother cell. Now, nucellus has towards this end is micropyle end, towards this end is chalaza end. It has one megaspore mother cell. This megaspore mother cell, diploid, undergoes reduction division and produces 4 haploid megaspores. In these 4 megaspores, 3 degenerate, that means disintegrate. If 3 degenerate, only 1 remain functional towards the micropylar end. So, this is functional megaspore. This functional megaspore undergoes, now this is important for MSET. Now, this undergoes 3 times mitotic division. First time mitotic division, it produces 2 nuclei. Second time, out of 2, both are present at the micropylar end, one slowly comes to chalaza end. Now, one is at the micropylar end, one is at the chalaza end. Now, second time mitosis division takes place, 4 nuclei are formed, 2 at the micropylar end, 2 at the chalaza end. Third time mitotic division takes place, 8 nuclei are formed, 4 at the micropylar end and 4 at the chalaza end. Now, how many times mitotic division takes place means 1, 2, 3, 4, 5, 6 and 7 times mitotic divisions takes place. 3 times mitotic division, 7 mitotic division. The upper are called upper 4 polar nuclei, lower are called lower 4 polar nuclei. In this, one comes to the center and ultimately the arrangement will be 3 plus 2 plus 3. Now, till now, free nuclear divisions takes place without any cytoplasmic division. Now, cytoplasmic division takes place and cells are formed. This will form an 8-celled or 7-celled 8 nucleated embryo sac or female gametophyte. In these 7-celled 8 nucleated embryo sac, upper 3 cells are called egg apparatus with 2 synergids and 1 egg cell. The central 2 are called polar nuclei. The lower 3 are called antipodals. So, now if you take this structure, it appears like this. Gradually, the 2 polar nuclei fuses and ultimately a structure like this, 7-celled, 7 nucleated embryo sac is formed. These 2 are called synergids. This is called egg cell. This is called secondary nucleus formed by the fusion of polar nuclei. This is called antipodals. Now, in the synergids, these two, synergids and egg cell combinedly called egg apparatus. Now, these two synergids contain special finger-shaped structures called filiform apparatus. Filiform apparatus perform two functions. One is they collect the food from nucellus and supply it to the embryo sac, developing embryo sac, and it also secretes the chemical substances which guides the movement of pollen tube into the ovule. Egg cell involves in syngamy and secondary nucleus involves in triple fusion. Like this, embryo sac is formed, and this is called polygonum type of embryo sac because it was reported in Polygonum divaricatum by Strasburger. It is also called monosporic embryo sac because it develops from a single functional megaspore. This is the development of embryo sac. Next, pollination. The transfer of pollen grains from anther to the stigma is called pollination. Pollination in plants is of two types: direct pollination, indirect pollination. Direct pollination means pollen grains first reach the ovules because here ovary, style and stigma are absent. When pollen grains first reach the ovule, it is called direct pollination found in gymnosperms. Sometimes in some plants, pollen grains first reach the stigma, then goes to the ovule. It is called indirect pollination, which is found in angiosperms. In indirect pollination of two types: cleistogamy, pollination in a completely closed flower which never open in its lifetime. Chasmogamy, pollination in an opened flower. Chasmogamy is of two types: autogamy, pollination in a single flower; allogamy, pollination in between two flowers. Allogamy is again two types: geitonogamy, xenogamy. Geitonogamy means pollination between two flowers of the same plant. Xenogamy means pollination between two plants of the same species. Geitonogamy is pollination between two flowers of the same plant. It is almost a self-pollination. Xenogamy means pollination in between two flowers of different plant but belonging to the same species. This is introduction to pollination. Then comes the contrivances of cross-pollination. Contrivance of cross-pollination means for a plant, cross-pollination is more beneficial than self-pollination. Because by cross-pollination, disease-resistant characters are formed, more number of seeds are formed, seeds show maximum vigor and vitality, and there are so many advantages. So, plants try to perform self-pollination, try to avoid self-pollination and perform cross-pollination. The contrivance of cross-pollination are dicliny, dichogamy, hercogamy, heterostyly, pollen prepotency, self-sterility and sensitive stigmas. They are all contrivances. If any one of that is present, the plant perform cross-pollination and avoid self-pollination. Dicliny is presence of unisexual flowers. Dichogamy is maturation of essential organs, andrecium and gynoecium at different times. Hercogamy means arrangement of essential organs in different heights or in different directions. Heterostyly means presence of different heights of styles in the flowers of the same plant. It may be diheterostyly or triheterostyly. Self-sterility means suppose the pollen grains reach the stigma of the same flower, they do not show any germination. Passiflora and abutilon and orchids. Then pollen prepotency, domination of cross-pollination over self-pollination. Sensitive stigmas, stigmas are very sensitive to the self-pollination, but they easily receive the pollen grains of other plants. Suppose cross-pollination fails, immediately plants perform self-pollination. For that they have some adaptations. The best example is Yucca gloriosa. The flowers of Yucca gloriosa shows symbiotic association with a special insect called Pronuba yuccacella. If that insect is there, the plant perform cross-pollination. If that insect is not there, the plant performs self-pollination. Homogamy, movement of floral parts; incomplete dichogamy, safety mechanism and cleistogamy are some of the things, but only three is given in our syllabus. One is homogamy. Presence of bisexual flowers and maturation of essential organs at the same time is homogamy, caltha and ranunculus. In these two plants, pollen grains are transferred from anther to stigma with the help of raindrops. Then safety mechanism. This is found only in the disc florets of Asteraceae. Here, the pollen grains, once the plant is protandrous, if cross-pollination fails, the stigmatic lobes bend downwards, touch the anthers and collects the pollen grains. This is called safety mechanism. And this safety mechanism is found in Asteraceae disc florets. Then cleistogamy. When a flower never open in its lifetime, there is no question of cross-pollination, there will be only self-pollination. Commelina bengalensis. Cleistogamous flowers are underground, chasmogamous flowers are aerial. Streptocarpus princeps, clistogamous flowers and chasmogamous flowers, both are aerial. This is about the contrivances of self-pollination. Then agents of pollination. Pollen grains cannot move by themselves because they do not have any flagella or cilia or whatever locomotive structures. So they have to be taken from one place to other place by some agents, some external things. There are two types of agents. One is non-living agents, the other is living agents. Abiotic components, biotic components. Abiotic components are air and water. Biotic component is animal. Pollination by air is anemophily. Pollination by water is hydrophily. If pollination occurs on the surface of the water, it is epihydrophily, Vallisneria. Under the water, it is hypohydrophily, it is Zostera. Then pollination by animals, zoophily. Pollination by birds, ornithophily, Sterlitzia, Bignonia. Pollination by bats, Chiropterophily, Chygelia pinnata, sausage tree. Pollination by insects, entomophily, Cestrum nocturnum. Pollination by snails and slugs, Melacophily, which is found in Aroidae plants, Colocasia and other plants. Now, this is about the explanation of two lessons, megasporogenesis and pollination. Now, we discuss some important questions in this chapter. So, first question is, choose the correct sentence from the following in the development of angiospermic embryo sac. Choose the correct sequence means what will form first, what will form next, and what is the sequence of the formation. Megaspore mother cell, archisporium, functional megaspore, embryo sac is wrong because first formed is archisporium. Archisporium, megaspore mother cell, functional megaspore, embryo sac is correct because archisporium divides and gives rise to megaspore mother cell. Megaspore mother cell divides by meiosis and produces 4 megaspores where 3 disintegrate, 1 remain functional. Then this megaspore undergoes numerous free nuclear divisions and later it produces cell wall resulting in the formation of embryo sac. So, the correct sequence from the following in the development of angiospermic embryo sac is archisporium, megaspore mother cell, functional megaspore, embryo sac. Next, only true cross-pollination occurs in Vallisneria. True cross-pollination means xenogamy. Geitonogamy cannot be regarded as true cross-pollination because in geitonogamy, the pollination occurs between two flowers of the same plant. It is not a cross-pollination, it is almost a self-pollination. Real cross-pollination is xenogamy. Why? This process occurs in between two flowers of different plant but belonging to the same species. In Vallisneria, self-pollination is not possible. Definitely in Vallisneria, the plants are dioecious. Dioecious means separate male plant and separate female plant. Whenever plants are dioecious, no question that a dicliny or unisexuality is observed. So, no question of self-pollination. So, only true cross-pollination occurs in Vallisneria is correct. In Vallisneria, self-pollination is not possible is correct. So, A and R are true. R does not explain the A because only true cross-pollination occurs in Vallisneria. If it is asked like that, Vallisneria plants are dioecious must be asked. But here a different statement is given, different assertion is given. So, answer is both A and R are correct. R is not the correct explanation of A. Now, coming to the matching. List 1 is Allium, Polygonum, Peperomia, Fritillaria. List 2 is monosporic, bisporic, tetrasporic, tetrasporic. Allium is always bisporic. So, for this the answer is 4. 1 that is Allium, B bisporic. Bisporic means out of 4 megaspores formed, 2 are disintegrate and 2 remain functional. Polygonum we all know monosporic. So, Polygonum 3 megaspores disintegrates and 1 megaspore is functional. Peperomia and Fritillaria are tetrasporic. That means all the 4 megaspores are functional and they produce a embryo sac. So, Allium, Polygonum, Peperomia, Fritillaria. Answer is B, A, C, D. Monosporic, bisporic, tetrasporic and tetrasporic. Now, assertion. Only xenogamy is possible in Boracious. Boracious is a dioecious plant. Whenever there is a dioecious plant, male plant and female plant are separate. Male plant shows only male flowers with androecium. Female plant shows only female flowers with gynoecium. No chance of self-pollination, only cross-pollination occurs, and that cross-pollination is also xenogamy. Pollination between two different flowers of the same plant belonging to the same species. So, only xenogamy is possible in Boracious is correct. Why only xylogamy is possible in Boraces? Because Boraces is a dioecious plant. So both A and R are correct. R is the correct explanation of A. This is the answer for this question. Now, triplet of cells found at the chelazal end of embryo sac. Triplet of cells means 3 cells. Triplet of cells present at the chelazal end of embryo sac. This is micropellar end and this is chelazal end. This is one triplet of cells. Triplet of cells means 3 cells. This is one central cell. There is a group of triplet of cells towards the chelazal end. These are antipodal. So antipodals are the triplet of cells present towards the chelazal end of embryo sac. So embryo sac shows 2 triplets of cells. Upper triplet is called egg apparatus with 2 synergids and 1 egg cell. Lower triplets are called antipodals. These antipodals disintegrate before fertilization or after fertilization. There is no perfect use of these antipodals. That's why these are popularly regarded as vegetative cells of embryo sac. Dichogamy, hercogamy and heterostyle all are found in the flowers of dichogamy, hercogamy, heterostyle all are present in the flowers of primula, because primula shows diheterostyle. Diheterostyle means presence of dimorphic flowers. Dimorphic flowers means 2 different types of flowers. A single plant shows some flowers with long styles and short stamens, some flowers with short stamens and long styles. Pollination always occurs between the flowers of long styles and long stamens, short styles and short stamens. There, the arrangement of essential organs is in different heights. So that is ergogamy. The maturation of essential organs, androecium and gynoecium, is at different times. So that is dichogamy. 2 different heights of styles present in the flowers of the same plant, that is heterostyle. So dichogamy, hercogamy and heterostyle all are present in the flowers of means primula. Primula shows diheterostyle. Now, arrange the following in their correct sequence of formation. How they are formed? For this, answer is 2, 4, 3, 1. First is synergids, 2 is archisporial cell, 3 is embryo sac, 4 is megaspore. So always in a ovule, within the nucellus or megasporangium, first formed is archisporial cell. Archisporial cell divides and produces megaspore mother cell. Megaspore mother cells by meiosis produce megaspore. So first formed is archisporial cell, then formed is megaspore, then is embryo sac, 7-celled 8-nucleated embryo sac or 7-celled 7-nucleated embryo sac, and in the embryo sac synergids are present. So what is the correct order? First archisporial cell, then megaspore, then embryo sac, then synergids. So answer is 2, 4, 3, 1. That is the correct sequence of formation of various cells during the development of embryo sac. Now this is a matching. Study the following list: Oldenlandia, Lythrum, Abutilon, Dolichus. This is regarding contrivances of cross pollination. Oldenlandia, Lythrum, Abutilon, Dolichus, and list 2 is self-sterility, pollen prepotency, diheterostyle, triheterostyle, sensitive stigmas. The answer is 3. A is 3. What is Oldenlandia? Diheterostyle. Just now I explained about primula. For diheterostyle, there are 2 examples, primula and Oldenlandia. B is 4. What is B? Lythrum. Lythrum shows triheterostyle. Triheterostyle means 3 different heights of styles: long styles, short styles, medium styles, long stamens, short stamens, medium stamens. So 3 different heights of styles are present in Lythrum and oxalis. Next, C, Abutilon. Abutilon is self-sterility. Abutilon and passiflora, even though bisexual flowers are present, self-pollination does not occur, only cross-pollination occur. Why? Because self-sterility. What is self-sterility? Self-sterility is when the pollen grains reach the stigma of the same flower, they do not show any germination. That is failure of self-pollination in a bisexual flower. Failure of self-pollination in a bisexual flower is self-sterility. What is self-sterility? When the pollen grains reach the stigma of the same flower, they do not show any germination. Suppose the same pollen grains reach the stigma of the other flower, they show germination and helps in fertilization. Then dolichos. Dolichos shows pollen prepotency. Pollen prepotency means domination of cross pollination over self pollination. That means in a flower, in a bisexual flower, stigma receives the pollen grains of same flower and pollen grains of other flower. The pollen grains of other flower matures, germinates much before than the pollen grains of same flower, that is called pollen prepotency. So Waldenlandia diheterostyly, Lythrum triheterostyly, Abutilon self-sterility, Dolichos pollen prepotency. Sensitive stigmas are found in Mimulus and Martinia. Sensitive stigmas are normally found in Mimulus and Martinia. The female gametophyte of a typical angiosperm at the time of fertilization. The female gametophyte of a typical angiosperm means embryo sac. The female gametophyte of a typical angiosperm means an embryo sac. Embryo sac at the time of fertilization show T types of cells. They are two synergids with filiform apparatus, one egg cell, both combinedly form egg apparatus, central secondary nucleus or central cell formed by the fusion of polar nuclei, and 3 antipodals. So it is 7-celled and 7-nucleated or 8-nucleated. 7-celled, 7-nucleated or 8-nucleated. 7-celled 1, 2, 3, 4, 5, 6, 7. Why 7-nucleated means 1, 2, 3, 4, 5, 6, 7. But this is diploid. Diploid because it is formed by the fusion of polar nuclei. So because it is diploid, it can be regarded as 2 nuclei or 1 nuclei. So answer is 7-celled and 7-nucleated or 8-nucleated while it is ready to perform or involve in fertilization. Now, xenogamy is a true cross pollination. Really, xenogamy is a true cross pollination because here pollen grains transfer from anther of one flower to the stigma of another flower present on different plant but belonging to the same species. Reason: xenogamy occurs between two different species and results in gene combinations. Xenogamy absolutely does not occur in between two different species. It occurs only in between two plants of same species. So here assertion is correct. Xenogamy is a true cross pollination is correct. Reason is wrong because xenogamy occurs between not two different species but it occurs in between two different plants belonging to the same species and results in gene combination. These are all wrong. So assertion is correct. Reason is false. A is true, R is false. Number of meiotic and mitotic divisions involved in the formation of polygonum type of embryo sac starting from megaspore mother cell respectively. Megaspore mother cell by one reduction division produces 4 megaspores. In these 4 megaspores, 3 degenerate. 1 megaspore remain functional. This megaspore undergoes 3 times mitosis or 7 mitotic division. By first mitosis 2, by second mitosis 4, by third mitosis 8, by third mitosis 8, like this. Out of 8, upper 4 are called upper polar nuclei, lower 4 are called lower polar nuclei. From upper polar nuclei 1, from lower polar nuclei 1 migrates to the center. Arrangement will be 3 plus 2 plus 3 arrangement. Cell wall formation takes place and ultimately embryo sac is formed. So the answer for this is 1 meiotic division and 7 mitotic divisions. 1 meiotic division and 7 mitotic division results in the development of polygonum type of embryo sac starting from megaspore mother cell. This is a 3-column tabular form. Study the following and identify the correct combination. Plant, pollination, type of contrivancy. Vallisneria, autogamy, dicliny is Vallisneria, autogamy, dicliny. Here you have to identify the correct combinations. Correct. Lathyrus, allogamy, triheterostyly. Lythrum, allogamy, triheterostyly. So answer is 1, 2, 3. Ranunculus, autogamy, homogamy. Caltha and ranunculus, always homogamy takes place. Argemone, allogamy, movement of floral parts. So that is wrong. So 1, 2, 3 are correct. Next, find out the correct statement among the following. Self-pollination cannot be possible in central flower of Cyathium. Xenogamy is possible in Vallisneria. Autogamy is possible in all the flowers of mango. Allogamy is not possible in central florets of Drydax. So here, find the correct statement among the first statement is the correct statement. Why? Because self-pollination cannot possible in central flower of Cyathium, because central flower of Cyathium is a female flower. It shows ovary, style and stigma and a jointed stalk. Ovary is tricarpillary, syncarpous, trilocular in nature. So it is a single female flower. It cannot show self-pollination. Xenogamy is possible in Vallisneria is correct because it is a dioecious plant. Autogamy is possible in all flowers of mango is also correct because bisexual flowers, female flowers and male flowers are present. Allogamy is not possible in the central florets of Asteraceae. Central florets of Asteraceae, allogamy may not be possible. Next, dikaryotic cell of embryo sac. Dikaryotic cell of embryo sac means a cell which shows two sets of chromosomes. For this, the answer is 4. Central cell, because synergid is haploid, monokaryotic cell. Egg cell is haploid, monokaryotic cell. Antipodal is haploid, monokaryotic cell. Only central cell which is formed by the fusion of two polar nuclei is the dikaryotic cell of embryo sac. Next, in angiosperm, free nuclear divisions occurs during. Free nuclear divisions means only nuclei are formed, no question of cytoplasm division takes place. Microspore formation is not a free nuclear division. Megaspore formation is not a free nuclear division. Flower formation is absolutely not a free nuclear division. The only answer for this is embryo sac formation. Why? Because megaspore mother cell undergo reduction division and produce 4 haploid nuclei. 3 nuclei disintegrate and 1 remain functional. This functional starts divisions. First it produces 2, then it produces 4, then it produces 8. Then movement of nuclei takes place. Then only cytoplasmic division takes place. So right from meiosis of megaspore mother cell to the arrangement of nuclei, no question of cytoplasmic division. Only nuclear division takes place. That's why in angiosperms free nuclear divisions occurs during means embryo sac formation. Flowers of clerodendron are structurally bisexual but functionally unisexual. The flowers of clerodendron are protandrous. Clerodendron means, we all know, clerodendron is a dicaecal sac. Peduncle grows definitely, ends as a flower. From the lower side, 2 lateral branches develop, they also become flowers. 2 lateral branches again produce 2 lateral branches which also become flowers. Again 2 lateral branches produces 2 lateral branches and they also become flowers. This is dicaecal sac. So in clerodendron, this is the type of inflorescence. Flowers of clerodendron are structurally bisexual but functionally unisexual. Structurally bisexual because they contain androecium and gynoecium, so they are definitely structurally bisexual. But functionally unisexual because they show protandry. Protandry means androecium matures much before gynoecium. This is the most common type of protandry in majority of the flowers, or dichogamy in majority of the flowers. Androecium because it matures much before and liberates the pollen grains. After the liberation of pollen grains only 7-celled 8-nucleated embryo sac is formed. So definitely flowers of clerodendron are structurally bisexual but functionally unisexual. The flowers of protandrous, because the flowers are protandrous, then only they are called functionally unisexual. So structurally bisexual, even though they have androecium and gynoecium, they cannot help in self-pollination. So A and R are correct. R is the correct explanation of A. In angiospermic ovule, the synergids are. In an angiospermic ovule, whenever the synergids come, they are only haploid. They are not diploid. Even though two are there, two are present separately. They are not diploid. They are not haploid before fertilization and diploid after fertilization, and they are not triploid. Triploid is only endosperm. Haploid before fertilization, diploid after fertilization is egg cell. Egg cell is haploid before fertilization and diploid after fertilization, because it will become zygote after fertilization by syngamy. So, the answer for this is haploid. In angiospermic ovules, synergids are always haploid. Pair of taxa that show sensitive stigmas to self pollen. Sensitive stigmas to self pollen, Mimulus martinia. The special feature in these two plants is whenever the anther of the same flower is liberating the pollen grains, they closes the sensitive areas in the stigma which receives the pollens and helps in fertilization. Because they closes the stigmatic lobes, they cannot collect the pollen grains liberated from the anther of the same flower. They cannot collect the pollen grain from the anther of the same flower. So, self-pollination is prevented. Once the total pollen of the same flower are liberated out, once the total pollen of self-pollinated pollen means same flower are liberated out, immediately the stigma becomes erect and makes the receptive portion to get the pollen of other flower, so that it performs cross-pollination and completes cross-pollination. So, Mimulus and Martinia are the pair of taxa that shows sensitive stigmas to self-pollen. Self-pollen, when it is liberated, they are completely closed. Once the total liberation of self-pollen takes place, immediately they starts opening the stigma, and once they open the stigma, cross-pollination takes place and plants will successfully avoid self-pollination and perform cross-pollination. Solanum scrofularia shows protogyny, maturation of gynoecium much before androecium. Primula oldenlandia shows diheterostyly, presence of two different heights of styles. Just now I told it is a combination of protandry, it is a combination of dichogamy, hercogamy and heterostyly. Abutilon passiflora is self-sterility. Self-sterility means the pollen grains that reach the stigma of the same flower cannot show germination. That means the pollen grains liberated from the anther of the flower, suppose reaches the stigma of the same flower, they do not show any germination. So, answer for this is Martinia and Mimulus. Arrange the following structures in ascending order based on the minimum number of meiotic divisions required for their formation. For this, you have to remember one. For the formation of one embryo sac, with two synergids, one egg cell, one secondary nucleus and three antipodals. The total number of meiotic divisions required is 1. This you have to remember. For producing two synergids, 1 meiosis. For producing one egg cell, 1 meiosis. For producing one secondary nucleus, 1 meiosis. For producing 3 antipodals, 1 meiosis. So, here 120 antipodals. 120 antipodals means for 3 antipodals 1. So, 40 meiotic. 120 antipodals means 40 meiotic. 60 synergids means 2. 30 meiotic. 2 synergids. Then 25 egg cells. 25 egg cells means 25 by 1. 25 meiotic. And 50 embryo sacs. 50 embryo sacs also by 1. 50 embryo sacs, 50 meiotic. So, here what is the question asked? Arrange the following structures in ascending order basis on minimum number of meiotic divisions required for their formation. Answer is 2. 25, 50, I do not say 25, 30, 40 and 50, 25. So, 3, 2, 1, 4. So, answer for this is 25, 3, 2, 1, 4. Now, Self-pollination is not possible in the flowers of Scrophularia. Self-pollination is not possible in the flowers of Scrophularia. Just now I told Scrophularia shows bisexual flowers with protogynous condition. Protogynous condition means it has to produce flowers bisexual, but gynoecium matures much before androecium. Flowers of Scrophularia are protandrous. So, self-pollination is not possible in the flowers of Scrophularia is correct because it is protogynous. Reason is wrong. Flowers of Scrophularia are not protandrous. Flowers of Scrophularia are protogynous. So, protogyny means maturation of gynoecium much before androecium. Self-pollination is not possible in the flowers of Scrophularia is very much correct. Very much correct because they are all protogynous. Flowers of Scrophularia are protandrous is wrong. Flowers of Scrophularia are not protandrous. Flowers of Scrophularia are protogynous. So, A is correct, R is false. This is about some of the important questions regarding development of embryo sac and pollination. Regarding development of embryo sac and pollination. Development of embryo sac and pollination. Now, next is after pollination, the next concept is fertilization and fruits. So, with this completion of fruits, we will complete the total morphology. Now, next concept we are going to discuss is fertilization and fruits. So, fertilization, syngamy by Strasburger, fusion of male gamete. The fertilization means we have to remember how pollen grains comes and reach the stigma. Stigma secrete chemical substances which absorbed by pollen grain which gives rise to pollen tubes. In majority of the plants, one pollen tube monosiphonous condition. In the plants of Malvaceae and Cucurbitaceae, polysiphonous condition, more than 2 or 3 or 4 pollen tubes. In the pollen tube, generative cell, vegetative cell enters, generative cell divides and produces 2 male gametes. Pollen tube with 2 male gametes enters the ovule either by porogamy, otelia. Mesogamy means integuments and funicle cucurbites. Chalazogamy means through chalaza, which was recently discovered in Casuarina plants by Troy. Any one of these methods, they enter the ovule by piercing through the synergids. Later, the tip of the pollen tube ruptures, subapical end of pollen tube ruptures or tip of the pollen tube dissolves and both the male gametes are released. When the male gametes are released, one male gamete fuses with the egg cell to form a zygote which is called syngamy, reported by Strasburger. The other male gamete fuses with the secondary nucleus to form a triploid primary endosperm nucleus which later develops into endosperm which is called as triple fusion, and this is reported by Navasin and in Lilium and Fritillaria beginning days, but later in almost all plants. This is fertilization which is popularly called double fertilization. Now, we discuss some of the important questions, some stock questions, some standard questions which are asked in the previous years in the MCET examinations regarding fertilization and fruits. Question number 1, assertion. Angiosperm endosperm cell shows 3 sets of chromosomes. The endosperm of angiosperm cell shows 3 sets of chromosomes. 2 sets of female parent and 1 set of male parent are found in endosperm. The answer is 1. Why? Because 3 sets of chromosomes, 2 sets of female parents. What are the 2 sets of female parents? Secondary nucleus. 1 set of male parent. What is the 1 set of male parent? Male gamete. So, 1 set of male parent, male gamete, 2 sets of female parent, secondary nucleus, fused to form a triploid primary endosperm nucleus which later slowly converts into endosperm which is triploid. So, angiospermic endosperm cell shows 3 sets of chromosomes is correct. 1 set of chromosome is called genome, 3 sets of chromosomes mean 3 genomes. 3 genomes is correct. Why 3 sets of chromosomes means 2 sets of female parent, that is secondary nucleus, 1 set of male parent, that is male gamete, fuses to form endosperm. So, both A and R are correct. R is the correct explanation of A. Now, study the following table. Pepo, unilocular inferior cucumis. Pepo, unilocular inferior cucumis. unilocular inferior cucumis. Drupe, bilocular inferior mangifera. Incorrect combinations we have to identify. Pepo, unilocular inferior cucumis is correct. Drupe, bilocular inferior mangifera, it is not inferior, it is superior. You have to remember this. Bilocular superior over. Hesperidium multilocular superior citrus. Hesperidium multilocular superior citrus. And Pome, pentalocular superior. Pentalocular is wrong. So here the wrong concepts are 2 and 4. 2 and 4 are wrong. Why? Because Pepo, unilocular inferior cucumis is correct. Drupe, bilocular inferior mangifera is wrong. It is not inferior, it is superior. Hesperidium multilocular superior citrus is correct. It is the correct combination. Pome, pentalocular superior pyrus. It is not pentalocular, it is trilocular. So pome is also said to be a false fruit. Why? Because pome is found in apple. In apple, the edible part is thalamus. The actual fruit is pericarp, which is very hard, present at the center and absolutely not edible. Endosperm is absent in the seeds of Orchidaceae. The Orchidaceae and Trapezae endosperm is absent. It does not mean that triple fusion does not occur in that. Triple fusion occurs, but the total endosperm is consumed. So endosperm is absent in the seeds of orchids is correct, but there is no triple fusion in orchids is wrong. Triple fusion occurs in orchids, endosperm is completely consumed. So A is correct, R is false. Endosperm is absent in the seeds of orchids, and why endosperm is absent in the seeds of orchids means the developing embryo completely utilizes the endosperm. So there is no triple fusion in orchids is wrong. So answer is A is true, R is false. Aggregate fruit develops from the apocarpus gynoecium of acyclic flower is found in. Acyclic flower is found in. Flowers are of 3 types: acyclic flowers, hemicyclic flowers, cyclic flower. Acyclic flower means androecium, gynoecium, calyx, corolla arranged in a spiral manner. Example, magnolia. Hemicyclic flower, calyx, corolla arranged in a cyclic manner, androecium, gynoecium, spiral manner. Annona, polyalthia. Calyx, corolla, androecium, gynoecium all are present in a cyclic manner, cyclic flower, mangifera and many dicots. In magnolia, the fruit is an etirio of follicles. So etirio of follicles means an aggregate fruit. Aggregate fruit etirio of follicles is formed from the apocarpus gynoecium of an acyclic flower means aggregate fruit develops from the apocarpus gynoecium of an acyclic flower is, here the answer is magnolia. Annona is etirio of berries. It is a hemicyclic flower. Polyalthia is same, etirio of berries. It is a hemicyclic flowers. Nelumbo is etirio of achenes, but it is not acyclic or cyclic. It is apocarpus condition. Now, study the following and identify the correct match. Study the following and identify the correct match. Structure before seed formation, structure after seed formation. List 1 is structure before seed formation. List 2 is structure after seed formation. Funiculus, scar of the ovule, zygote, inner integument, hilum, tegmen, testa, stalk of the seed, embryo. Funiculus, stalk of the seed. So A is 4. Scar of the ovule, hilum. A permanent scar present on the ovule is called hilum. What is hilum? The place where funicle is attached to the ovule is called hilum. This hilum remains as a permanent scar in the seed. A dry fruit and dry seed is given to you. How can you say that one is a fruit, other is seed? In fruit you don't find hilum. In seed you will find hilum. Hilum is the scar present on the ovule. Zygote. Zygote develops into embryo. Zygote develops into embryo. then inner integument becomes tegmen, outer integument becomes testa. Inner integument normally become tegmen, inner integument becomes tegmen, outer integument become testa. So answer funiculus, stalk of the seed, scar of the ovule, hilum, zygote, embryo, and inner integument is tegmen. Next.